had one question from Zoom asking, what is the targeted timeline for completion of the different trials in human cells?
Well, so as far as the haploinsufficient lines, this is something we're very new at as a lab. So it's going to be, number one, differentiating them to the various cell types, and that does take time. And number two, then being able to test all the different treatment modalities. So as a specific timeline, it's going to be very hard to pin down, but we're working hard on it right now. And we just got these cell lines about a month and a half ago. And so these are all very, very preliminary pilot studies.
Got another one from online as well, just asking, kind of in your opinion, what assets, tools, or studies might be lacking currently, and where should we be kind of focusing?
I think right now, I can't think of anything we're absolutely lacking other than probably money, people, time.
That's the next question.
I was going to say.
John spent the last 21 days in the lab every day, so he wants more people. I think we have the ideas. I think we have what we need. What we really need is, I would love to get some kind of major backing, some angel investors and pharmaceutical company that would help us do this so that we could get, all the toxicology studies that we need and really make progress on what we've accomplished so far. Because I feel like we have the beginnings of something really important and we just need to keep going with it.
As someone who I have no idea how much money that would be, like how much what would you ask for from a big donor?
From the research or to move it to a clinical trial?
So clinical trial with the medicine.
What I've been quoted, because I'm not expert at this, but I've talked to a lot of people who have done this in their lives, Gary Cox, Wendy Chung, and other people from major pharmaceutical companies. Toxicology studies can cost them alone, can cost about $1 million. And then when you go past the toxicology studies to starting the clinical trials, it's a couple of million.
At that point in time, it's usually a pharmaceutical or biotech.
We are trying to engage pharmaceutical companies, large pharmaceutical companies that would be interested because some of them have worked with some of these molecules because of looking at it for Alzheimer's.
Yeah, I think Michelle's approached, 5 different pharmaceutical companies in the last 2 months. So
Do the human, what do you test first? We're testing in humans, do you test from blood products or do you test an actual human? Like what do you test for mice to like? I'm just really curious about that.
The blood samples that your families are donating to Simon's, those blood cells, there are cells that are taken from there, white blood cells that are then programmed to de-differentiate, to become pluripotent. And the value then is that they can be then differentiated to any cell type. And so Simon's is taking them and generating, Wendy talked about it, what we call these human-induced pluripotent stem cells. They can be turned into what we're going to do, which is brain excitatory neurons. We could also do inhibitory neurons. We could do glia, but we're going to start with the excitatory neurons, spinal motor neurons, and muscle cells. And so you can differentiate them to these different cell types and then test the different treatment therapies. So we have selected, I think we're starting with about 6, and we might add a few more where we took de-identified mutations from the Simon's collection that look like they're going across the whole gene. They're different mutation types because we really want to know, does the treatment work? Which of the different treatments works best, and are they safe? We want to make sure there are no adverse effects. There might be certain mutations where there's a partial product. We don't want to increase the level of that partial product, and if one of these therapies increases that and it doesn't improve the phenotypes, that would be advised then to go ahead with that treatment with someone with that mutation. So we really are trying to be very cautious and very thorough.
mentioned you have a paper coming out, I think you said tomorrow.
We submitted it yesterday.
It hasn't been accepted.
Okay.
It's just been.
We're good with publishing it tomorrow day.
Yeah, I know.
No, I was just wondering, when do you expect to know about that, when, with your review process, and can you tell us what the name of the paper is so that we can be looking for it?
We'll share the paper when, the name, it's basically, typically it takes 1 to 2 months to get your reviews back. It's rare to get it with no revisions, which basically usually means a few more experiments. So these days, we all consider a paper to require about a year to actually be in press, but you know, maybe we'll be lucky. It's happened once.
Just a follow-up question to this, is there gonna be a preprint available on BioRx or something?
As soon as it's in press, we'll make it available to you guys.
Michelle, there's another question on Zoom, which I know we discussed about lithium, which I know you studied for a different disorder, and why are we not looking to that further?
So lithium is one of these interesting drugs. It increases beta-catenin levels, and partly it does it through GSK3. But lithium is what we in the field would call a dirty drug. It's much less specific than the things we're working with. It has a lot of other targets. GSK3 has other substrates besides beta-catenin. I don't want to say that it's specific only to that, but we're taking one that has such high specificity. It's been tested against 350 other human kinases, which is the best specificity out there. It's really a very nice molecule. Lithium, on the other hand, doesn't only affect GSK, it has other things that it affects. And it's been in use for bipolar disorder for decades. People can be on it really long term. So in that case, you might consider it relatively safe, but it does cause potential for thyroid and kidney problems, and it is not very well tolerated in children. And so we wanted to do, in the beginning, we thought we would test lithium, but we really wanted to do something that we felt was much safer and we'd be more comfortable with.
You mentioned gene therapy briefly. I guess I'm just thinking about, let's say in the future, drug therapy becomes available, and then you dropped it, that gene therapy becomes available. And you kind of mentioned it's possible. Could there be any conflict, or like, if you did the drug therapy, would that disqualify you from gene therapy?
As far as a clinical trial, I mean, again, we're not experts on clinical trials, but the nice thing about the small molecule, as Jerry mentioned, is that you can always stop it. At least this particular compound itself has a relatively short half-life. So in fact, we showed you the behavioral data in the mouse and how it actually rescued behavior sort of the 24-hour probe trial from fear conditioning. However, when we look longer term out, it's back down to the levels of the normal heterozygous mouse. So it's not a long lasting effect. It's at this point, it's something you would have to be considering taking on a possibly daily basis.
If a lot of the children here are relatively young, so looking far out, and then if any of these therapies come to fruition, and these kids are now 17, 18, 20, will they work? Will they give them back all those years that they lost, or by that time, is it something that that's just how their bodies would be?
It's a good question. It's honestly at this point, we just don't know. And what we've seen, at least in the mouse, is we treated these mice at what we call young adult stage for mice. And it did have a corrective effect. Whether it would be better starting younger, and that could also have its own potential for problems itself. We just don't know at this point, and it's something we're obviously looking to study.
I'm kind of optimistic that we have a pretty good window for treating at older ages, partly because of the results of the mouse and the fact that The beta-catenin function and the sodium potassium ATPase function, those are activity dependent, constantly changing, really important for plasticity. And that's relevant throughout all of life. And studies have been done like in aged mice and little pilots in humans where you increase beta-catenin in an aged mouse that has started to show some kind of decline, or an Alzheimer's model, it improves learning, even in very aged animals. And so that's something, will it go to completely normal? I can tell you, I would expect statistically significant improvements.
And like on a sort of separate point to that also, it's generally thought of that the younger the intervention, the better. This is something we're obviously going to have to test. But like you mentioned, a lot of your kids are quite young, but you're also probably doing the right things now that you know, like physical therapy, basically helping them out. Those kind of things make huge improvements in their life, and obviously, like, keep it up. You guys are doing a great job.
Yeah, activity increases beta-catenin levels. Keep them active. Don't wear them out. Don't let them wear you out. Keep them moving.
Mine wasn't really a question, so I just, before you had left, I just wanted to thank you for your time in the lab, like you said, you were there the last couple of days, and you tried to spend some time to interview, so I just want to thank you for everything that you guys have done for.
You guys inspire us and keep us going. And every time we sort of, hit some wall, we're like, okay, we gotta just do this anyway. Let's keep going. It's been great. So it's a shared goal.
One more question that came in on Zoom, and I think we don't know the answer to this yet, is how are you thinking this could potentially be delivered? Would it be injection, pill form, or is that something as we get further down the line?
We're hoping to modify it to be oral. Right now it's injection, but that's one of our goals is to make it oral because it would be just more readily administered that way. So that's one of the things we're going to move to.
I just wanted to, the point that you made quickly, and thanks for everyone glossed over. You said activity proves big. Is there any indication in over the years that there's any natural supplemental dietary things that increase beta-catenin?
Not really. I think, obviously good health, but yeah, physical activity. It gets into the sort of the details of the science. We talked about this Wnt pathway. Wnt it's a solid. Going to be a scientist, but I think you can follow what I'm talking about. Wnt is a soluble molecule released with activity. And it binds to these receptors on the cell, and it naturally inhibits GSK3. That's how we came to thinking about GSK3, because you've got this endogenous molecule that's always regulating beta-catenin levels. There are other molecules in the cell that also do it. But Wnt is activity dependent. And so, learning, memory, stimulation, physical activity, all of that. That's how we all improve our cognitive function and our skill set. And so part of that is just naturally increasing the beta-catenin. And I see changes. Like I was noting, when I went to the first fundraiser, I met Declan. That was summer of 2019. I see him from then to now. It's beautiful to see how much better he's talking, walking, interacting. So I'm very optimistic that it's keeping your kids active and hopefully getting a treatment in place. Things can be really great for all of the children.
Actually, on a personal note, actually, my niece has a disease called Rett syndrome. It's actually very, very similar in some ways to CTNNB1 syndrome. And obviously, my sister and her husband have been great advocates for her. And the physical therapy, skiing, horseback riding, it's improved her so much. So keep it up.